Good morning, Oya. Good morning, Sunita. I think Oya, you missed yesterday class, right? Are you there in yesterday class? Okay. If you are not there, please kindly watch the recorded. Don't forget it. Okay. Good morning, Saima. Good morning, Sadguri. Good morning, all. Okay. Good morning, Shweta. Okay, so friends, in the yesterday class, we have started secondary metabolites. When we started the secondary metabolites, we started understanding about different type of secondary metabolites. Okay, no problem. That's good. Oh yeah, that's good, good, good. Good morning, Priya. Good morning. Okay, so there are many secondary metabolites that we have studied, friends. One we call terpenes, another one we call phenols, and another one called, we friends, nitrogen containing compounds. And if everyone quickly recall, in the yesterday class we have completed the terpenes and we were reading about the phenols. This concept is all about remembering the maximum amount of information, friends. Please kindly don't forget any information. So friends, terpenes can be biosynthesized by two important pathways. One we called, friends, mevalonic acid pathway. Then friends, followed by the another one we can call as MEP pathway. So I hope all of you remember. And I told you, friends, remember the different type of terpenes along with its carbon number. So if you all people quickly recall, we discussed, right, GPP, which is a monoterpene, 10 carbon. This is very important to remember, friends. FPP, pharencyl biphosphate or pharencyl diphosphate, which is how many carbon, friends? 15 carbon. GGPP, ziranyl diphosphate. How many carbons, friends? Ziranyl diphosphate is the 20 carbon containing compound. Good morning, Jayashree. Please continue. Kindly don't forget the number of carbons and the type of terpenes, friends. They are very, very important. Okay, I hope all the students remember. We discussed five type of terpenes which are acting which are acting against the insect, and we read that two type of terpenes which are acting against the herbivores. So one is the pyrethroids, another one is the monoterpene, third one is the essential oil, fourth one is the limnoid, fifth one is the phytoexdizones. Please kindly don't forget. If you people quickly recall, I told you, right, phytoexdizones are the one which are similar with the insect molting hormones. Don't forget that, friends. And we read there are two type of terpene again is the herbivore. One we call cardinolites, another one we call, friends, saponins. Kindly don't forget the statement at all. So friends, phenols, what you have to understand by this word phenols, hope you remember. Okay, it's a very simple structure. I told you, friends, in the entire secondary metabolite, the largest class of the secondary metabolites is undoubtedly terpenes only. After the terpenes, the largest class of secondary metabolites is what, friends? Phenolics. Good morning, Sumaya. Continue. What are phenolics? It's a benzene ring with the OH group. That is what exactly we can call as phenols. And friends, what are the various pathways of phenols? We discussed, right, there are two pathways are there which are involved in the biosynthesis of phenol. One we called, friends, shikimic acid pathway, and friends, another one we called malonic acid pathway. If all of you very quickly recall my words in the yesterday whatever we discussed, I told you na, shikimic acid pathway, which is already asked a direct part B question also, I said yesterday, right? So friends, shikimic acid pathway is the major plant pathway. And what about the malonic acid pathway? By using malonic acid pathway also we can synthesize the phenols. There is no doubt about it. But the point is what? This particular malonic acid pathway never occur more significantly in the plant body. the malonic acid pathway occur abundantly in the fungus. Don't forget that. Every one of you listen to me very carefully, friends. So shikimic acid pathway is the major pathway in the synthesis of the plant phenolic. Please kindly don't forget. And friends, why we have given the name shikimic acid pathway? Friends, we have given the name shikimic acid because, friends, it is formed as an intermediate molecule. That is the reason we are calling it as what, friends? Shikimic acid. Please kindly don't forget it. And I hope all of you people remember, this is also I have highlighted in the yesterday class. If you very quickly recall, if we want to synthesize the terpene, first we have to synthesize an important precursor molecule. What is that precursor molecule? I told you, IPP. Once we synthesize the IPP, friends, we can easily synthesize the terpenes. Similarly, friends, if we want to synthesize phenols, we require a starting precursor. So friends, every one of you please kindly remember the statements. If we want to synthesize the phenols, the starting compound here required is the amino acid phenylalanine, which is an aromatic amino acid. Good morning, Gurunarani. Continue. So we require what, friends? Phenylalanine. Please kindly don't forget this. Okay, now I want you people to remember, if the shikimic acid pathway wants to occur, we require majorly two type of precursor. Is it? One we call it as a erythrose 4-phosphate. Another one we call, friends, phosphoenol pyruvic acid. These are the two major precursors that we require for the shikimic acid pathway. So these two precursors undergo the shikimic acid pathway. Ultimately, they are going to give rise to the phenylalanine. So friends, it's a kind of a rigid topic, as I told you yesterday. Just be patient, I will explain. Okay, even though you can't see the structures, no problem at all in this slide. You no need to remember any structures. Let me tell you clearly what exactly you have to read and remember. I will explain twice. Just listen to me carefully. Once you download the PPT, you can see the structures easily. Phenylalanine. This is what, friends? Phenylalanine is the one which is acting as a starting component for the biosynthesis of phenol. Friends, the phenylalanine can be treated by what? Can be treated by an enzyme called as Pol. Everyone listen, friends. Phenylalanine can be treated by an enzyme called as a Pol enzyme. Upon treating with the Pol enzyme, all of you listen to me. What the Pol enzyme will do? It remove the amine group. Friends, the concept is very simple. Phenylalanine having a amine group also, carboxyl group also. So that amine group can be removed by this particular Pol enzyme. Upon removing the amine group, Pol can be converted into transaminamic acid. Now friends, this particular transaminamic acid can be converted into coumaric acid. Very simple, my simple point, friends. Coumaric acid, upon treated by the CoA, it is converted into coumaryl CoA. Last and final, the coumaryl CoA can be treated by an enzyme. Friends, you no need to remember the structure. By an enzyme called as chalcone synthase, it is going to give rise to the chalcones. Chalcones are the one which is going to give rise to the different types of phenol. What are the different type of phenols? These are lignin, flavonoids, tannins. These are all derived from where, friends? These are all derived from this particular chalcones. I'll quickly recall this. Everyone look into this. Whomever did not understand, no need to remember the structure. Phenylalanine is the amino acid which is acting as the precursor for the biosynthesis of phenols. Phenylalanine can be treated by an enzyme called as a Pol enzyme. Upon treating with the Paul enzyme, phenylalanine converted into transaminamic acid. Transaminamic acid converted into coumaric acid. Coumaric acid, upon treating with the CoA, it is converted into coumaryl CoA. Coumaryl CoA, upon treating with an enzyme called as chalcone synthase, it is converted into chalcones. So friends, then the chalcones can give rise to the different type of phenols. These are all the different type of phenols. So you people no need to remember the structure, remember the schematic presentation. Majorly remember, precursor is the phenylalanine, enzyme is the Paul. Good morning Shivani, continue. Okay, now listen to me very carefully. What are the type of phenols? So friends, even in our 2022 exam, along with the terpenes, we got the question even from the phenol also. What you have to remember and read here, friends, you have to remember and read a lot of information about each and every class of the phenols. Okay, lot of information to be remembered. Focus, and it's easy and it's interesting. But you have to remember the statements. Phenols are the second largest. It's not the first largest. Phenols are the second largest in case of a secondary metabolites. All the time the largest type of secondary metabolites are terpenes only. Phenols are broadly categorized into, friends, lignins, flavonoids, tannins. Listen to me, friends. In the phenols, I told you after the terpenes, phenols are the largest class. In the phenols, lignin, flavonoid and tannins are present. Flavonoid is the largest class of phenol, which is a 15 carbon containing compound. And friends, these flavonoids are further divided into anthocyanin, which are colored pigments. Flavones are flavonols, isoflavones or isoflavonoids. These are all the various classes of what, friends? Flavonoids. So please kindly don't forget it. Now friends, we are going to read one by one elaboratively. Okay, let me explain you 15 minutes continuously. After that, I will give the time. You can ask all your questions. Okay, listen to me carefully. Lignins, what you have to remember by the lignin? Every one of you understand, friends. Lignin is the highly branched polymer of phenyl propanoid group. Once again, you no need to remember the structure. I will tell you certain structures you have to be remembered in the secondary metabolites. I will tell you what structures they are. So, lignins are the highly branched polymer of phenyl propanoid groups. If you all quickly recall why the plant is very stronger, because of its cell wall, we all people know that. And why the cell wall is very stronger? Just because of the presence of the cellulose. I hope every one of you remember, we read in our 10th standard also, cellulose is said to be the most abundant organic compound in the plant body. We all people commonly know that. I want you to remember, after the cellulose, listen to me friends, after the cellulose, the toughest organic compound in the plant body is the lignin only. Found in the cell wall of plants, most abundant organic compound after the cellulose in the plants. Okay, now please kindly look into the functions. They present in the cell walls, notably of the xylem tracheids and vessel elements, providing the strength and preventing the collapsing of vascular tissue. What do you mean by that? How can we explain this particular point? Friends, you all people know very well. Suppose example, this is the xylem tracheid elements and this is the xylem vessel elements. You all people know, right? The tracheid elements and vessel elements are the one which are responsible for the transport of the water. We all know very well. So friends, listen to me. Suppose example, when the water flow become more, all the students listen to me. When the water flow become more, sometimes what happen, there is a chance that xylem vessels and the tracheal elements, they may collapse, they may punctured. My friends, when they get collapsed or when they get punctured, immediately what is going to happen, friends? The water is getting leaked out. Is it or not? Once the xylem trachea elements or vessel elements get punctured, water come out. So friends, this particular lignin will provide mechanical strength to the vessel elements and tracheal elements. By that, friends, vessel elements and tracheal elements, they never ever break. What, my friends? The vessel elements and the tracheal elements, they never ever break because of this presence of lignin. And the second one, friends, once again I am reminding you, the topic is a kind of a rigid topic, lot of things to be remembered. So I really appreciate your patience when you are learning this particular concept. Next point, physical toughness prevents the animal feeding on the plant. Friends, how can I justify this particular statement? Physical toughness prevents the animal feeding on the plants. I told you, friends, you might observe some stems are very, very thin stems. Even though they are very thin, very strong they are. What herbivore like a goat and cow will do, especially goat, if you find any thin stem, it will try to break it. But it cannot break because even though stem is very thin, it's very, very stronger. So that's why how it is very stronger, friends, just because of the physical toughness. That physical toughness only can be provided by the lignin. Even though, friends, even though any herbivore or a pathogenic organism, when they start consuming the lignin from the plant, obviously, friends, lignin will be relatively indigestible. As a result, it can completely block or friends, it will completely inhibit the pathogenic growth. Please kindly do not forget the statement at all. Okay, this is the first one, lignins, and the next one, flavonoids. So what do you have to remember by the flavonoids? Flavonoids are the largest group of phenols. This is the direct question asked in our CSIR also. What is the statement they have asked? They asked that flavonoids are the 20 carbon containing compound, which is absolutely a wrong statement. All the students listen, friends. Flavonoids are the 15 carbon containing compound which consist of 2 aromatic rings which are linked via 3 carbon bridge. Every student, please kindly look into this. This is 2 carbon bridges which are connected by an intermediate. You can see, right? Very, very simple, na? So 6 plus 6 plus 3. So 2 aromatic rings connected by a 3 carbon bridge. That is what exactly we can call, friends, flavonoids. Every student remember all the, see, among the phenols, friends, flavonoids is the very, very, very largest class. So they are broadly categorized into anthocyanins, flavones or flavonols, then, friends, followed by the isoflavones. These three are the very largest class. Please kindly don't forget it. Now let's try to understand one by one. Isoflavones, we can also call it as isoflavonoids. Friends, the three classes are extremely important, very easy to remember and very important also for the exam. So let's talk about the first one, anthocyanin. I will tell you which structures to be remembered. Friends, anthocyanin is also a colored pigment and they are not responsible for the normal green color, right? So the green color can be, the green color can be mediated by what? Chlorophyll pigments. Chlorophyll pigments are responsible for the green color. Please listen, friends. Anthocyanins are, I will ask this in the tomorrow class also. Anthocyanins are exclusively responsible for blue color, red color, even purple colors also. Correct, na, friends? From the long distance, when the insect see these colors, insect will get attracted. They will go near that particular plant and they can start the process of pollination. So remember, anthocyanins are majorly responsible for blue, red as well as, friends, purple colors. Kindly don't forget it. And friends, here comes the unique statement. Look into the structure. You no need to remember, friends, but just look into this. Anthocyanin contain 3 aromatic rings. Correct na? You know, friends, what do you call anthocyanins here? We can call it glycosides. Don't forget, friends, whichever I will highlight, never ever forget. Anthocyanins are glycosides, where, my friends, sugar attached at position 3. What attached at position 3? Sugar attached at the position 3. When the sugar attached at the position 3, then only, friends, we called it as the anthocyanin. Please never forget this particular statement at all. Next one, friends, anthocyanidin. What is the difference between the anthocyanin and anthocyanidin? Here also 3 aromatic rings, here also 3 aromatic rings. But friends, in case of a anthocyanidin, at the 3rd position, sugar was not there. What is there? If any other group is present apart from the sugar, that we cannot call it as the anthocyanin. Now you can see here, friends, we will be calling it as the anthocyanidin. Never forget that. Look into this. Anthocyanins are the glycosides, where the sugar attached at the position 3. Never forget this point, friends. And what are anthocyanidins? Anthocyanidins, nothing but they are the anthocyanins without sugar. I told you, right? Instead of sugar, something else will be associated. Here, what is associated instead of the sugar? OH group is present. Very, very simple point. That is the anthocyanin. Now, what is the second class? Flavones and flavonols. Flavones and flavonols, isoflavones and isoflavonoids are very important. Please listen to me very carefully. Friends, listen to me. I am a plant. Just listen to me. I am a plant. Inside my body, flavones and flavonols are present. You know, friends, what is the speciality of the flavones and flavonols? Listen to me carefully. They can easily absorb the light of very shorter wavelength. Very shorter wavelength of light also can be very easily absorbed by this particular flavones and flavonols. Every one of you listen, my friends. They are absorbing the very shorter wavelength. Do you think they are visible to the human eye? Not at all. Since they are absorbing the light of very shorter wavelength, they are not at all visible to the human eye. Always remember that. Okay, the point is clear. They are not visible to the human eye. Flavones and flavonols. Then who can observe that? Who can perceive that? It can be perceived by, understand, friends, it can be perceived by insects. What is the speciality of the insects? Insects, friends, they can absorb the light in the UV spectrum very easily. Very easily. And serving as the attractants for pollinators. Right. And every student, please try to understand this particular important point. All the students try to understand. Flavonols also helpful in the pollination. How does the flavones or flavonols helpful in the pollination? By the formation of a nectar guides. What do you call it as? Nectar guides. Friends, what are these particular nectar guides? Symmetric pattern of stripes, concentric circle, or are spots. What do you mean by that? Every student try to understand. Look into this, friends. This is the leaf. This is the leaf. You know, in the leaf, there are different type of structures are there. We can call symmetric pattern of stripes like this. We can call symmetric pattern of stripes and concentric circles like this. Concentric circles. Concentric circles. Or are spots like this. Spots. Okay. You know, friends, all are very highly colored. By looking at this particular color, the insects can easily attract towards it and they will start the process of pollination. Very, very simple. That is what I am highlighting. What I am saying, flavonols are helpful in the pollination. How? Flavonols are helpful in the pollination by the formation of the nectar guides. Best example, symmetric pattern of stripes, concentric circles, as well as the spots. Kindly don't forget it. Now let me tell you one of the very interesting point about the flavones and flavonol. Flavones and flavonols plays a very crucial role in protecting the plant from the high UVB damage. High UVB damage. Friends, this is a very important point. Imagine, I am a plant body. Inside my body, what are there? Flavones and flavonols are there. You know what the flavones and flavonols present in my leaf? I am a plant. I am having leaves. In my leaves, flavones and flavonols are there. What they will do? They will absorb the UVB light. But here comes very interesting point. That is, I am a plant body. I can't survive. I can't survive without the process of photosynthesis. That's a common point. I can't survive without the process of photosynthesis. So if I want to perform the photosynthesis, I compulsory required what? I will be requiring the visible light. So every student has to remember, friends, this particular flavones and flavonols only absorb the UV light. It never ever interrupt visible light. Visible light can be absorbed by the plant body continuously for its all necessary functions. But flavones and flavonols never allow the plant to absorb the dangerous UVB light. Very, very simple point it is. It is present in the epidermis of leaves, and what they are absorbing, friends? They are absorbing the UVB light. They will absorb the UVB light, but friends, flavones and flavonols never ever interrupt visible light. They will allow the visible light to pass uninterruptedly. Please kindly do not forget this point. Now listen to me carefully, friends. Flavones and flavonols secreted into the soil by the leguminaceae plants mediated by the nitrogen fixation. I hope every student remember this without fail. While we are discussing the nitrogen metabolism, we clearly understood. How does plant root hair attract the bacteria? I hope every student remember, friends. Plant root hair, in order to attract the bacteria, hope you remember, it is basically releasing the chemoattractants. Yes or no? Of course, yes. Friends, from where, what are that particular chemoattractants? Best example, you can take a flavones, flavonols, and another example we can call the betaines. I hope you all people remembered, right? So these are all the chemoattractants, flavones, flavonols, and betaines released by the plant root hair into the soil for for attracting the bacteria to come near the root hair and start the process of nitrogen fixation. Please kindly do not forget it. Let me know if you have any kind of a doubts. Now the last class, last class of the flavonoids. Isoflavones and isoflavonoid. Friends, every one of you look into this carefully. Flavones and flavonols are having 3 aromatic rings, no doubt about it. Every student listen to me. Isoflavones or isoflavonoids also, friends, having 3 aromatic rings only, there is no doubt about it, in which one aromatic ring shifted its position. Correct now? You only try to understand. Here aromatic ring is in the upper position. Correct now? In the flavones and flavonols. Here the aromatic ring is shifted its position towards the downwards. You no need to remember the structure. So what are the best examples of the isoflavones or isoflavonoids present in the plant body? The best example, rotinoids, phytolexin. Now friends, you know the specialty of rotinoid. It is a type of isoflavone which is having a powerful insecticidal activity, which means whatever the plant, listen to me friends very carefully, whatever the plant that is having more rotinoids, they can easily fight against the insects. That's what it means. And whatever the plant that is having a phytolexin, which are the type of isoflavone or isoflavonoid, it will possess antimicrobial properties, which means that which can act on the bacterial infection as well as the fungal infection. My friends, even certain isoflavonoid will possess anti-cancer properties also. You might read many times, doctors and everyone will suggest us, drink more green tea. What happens when we drink more green tea? More in the sense sufficient amount, at least 1 or 2 cups of green tea every day. What happens when we drink a sufficient amount of a green tea? Friends, green tea leaves basically contain isoflavones or isoflavonoid. What they will do, friends, they will absorb the dangerous UV light, and they never ever allow, listen my words, they never ever allow the UV rays, dangerous UVB rays, to pass through the skin surface. You people commonly know, right? We discussed in the DNA repair mechanisms very clearly, friends. In the damages we study, right? When two thymine molecule associated with each other, what is happening, friends? We read, right? There is a formation of a pyrimidine dimer, and I hope all of you remember the pyrimidine dimers can lead to a dangerous disease, which is melanoma, which is a type of a skin cancer. Yes or no? Of course yes. So isoflavones or the isoflavonoids, they basically protect us from this particular melanomas or skin cancer, because they never allow the direct absorption of the UVB light by the skin surface. They never allow the UVB light to pass through the skin surface. Always remember that. Okay, so friends, so far what we studied in this particular phenols. So friends, we have studied lignins, flavonoids, and we also studied the 3 type of the flavonoids also. Then the last class of the phenols is what, friends? Tannins. Okay, let's talk about the tannins elaboratively. What are tannins? Same, tannins are also phenol. See, you have to remember 2 common points. What are the 2 common points? Every secondary metabolite, listen to me friends carefully, every secondary metabolite present in the plant body can be eaten up by the herbivore. That's why we can call every secondary metabolite is the feeding deterrent. What do you call? Feeding deterrent, which means which can be eaten up by this particular herbivore. And what is the second common or most common point? Every secondary metabolite having the defensive property. That's the most common statement. Same way, friends, plant phenolics also having a great defensive properties. And friends, every one of you kindly listen my statements. There are different type of tannins are there, friends. Many types, many types of the tannins are there. I can say, friends, more than 15 to 20 different type of tannins are there. But all those tannins, they are broadly categorized into 2 types. One we called condensed tannin, another one we called, friends, hydrolyzable tannin. Friends, let me explain you the clear-cut differences between the condensed tannins as well as, friends, hydrolyzable tannins. What are the differences? How can we differentiate them? All the students listen to me. Condensed tannins are formed by the polymerization of the flavonoid units. Always listen my statements very carefully, friends. They are formed by the polymerization of flavonoid unit. This is a flavonoid. See my friends, listen to me very carefully, friends. Condensed tannins formed by the polymerization of the same flavonoid. This is one flavonoid, another flavonoid, another flavonoid. All are same, right? So listen, all the flavonoids come close together, they can form the condensed tannin. Friends, everyone listen my statement. All the flavonoid units are of same type. You're getting my point? This is the same monomer, this is the same monomer, this is the same monomer. All group of monomers, listen to me, group of same monomers come close together, they can form this particular condensed tannin. Friends, since all the type of monomers are of same type, it is very difficult. It is very difficult to separate this particular condensed tannin. So if you want to separate this condensed tannin, my friends, we should always use the strong acids only. Kindly don't forget it. Condensed tannins, they are formed by the polymerization of the flavonoid units. They can also call it as proanthocyanidin. Friends, why do we call it them as a proanthocyanidin? Because they can easily hydrolyze it to anthocyanidin. Pro means inactive, anthocyanidin means active. How? By treatment with a strong acid. But friends, all of you kindly understand about the hydrolyzable tannins. Listen to me carefully. Hydrolyzable tannins are not formed by the same monomer. It is formed by the different monomer. That's why, you know, what do you call this particular hydrolyzable tannin? We call it the hydrolyzable tannin as the heterogeneous polymer. What do you call, my friends? We can call it heterogeneous polymer. Never ever forget the statement at all. See, look into this. This is the flavonoid, and along with that, what it is associated? It is associated with certain sugars like gallic acid. What I would like to say? I would like to say hydrolyzable tannin not made up of a same monomer. You don't need to remember the structure. It made up of a flavonoid plus a sugar called as gallic acid. Friends, listen my words carefully. Listen to me carefully. Since hydrolyzable tannin is made up of two different subunits. This is a different subunit, this is different subunit. Both are associated. But you know, this both interaction is the weaker interaction. So I can easily dissociate both of them even by using the dilute acids also. By using dilute acids also, friends, I can easily break this particular hydrolyzable tannins. Friends, but when you look into the condensed tannin, what did I tell you? Since they are made up of a same monomeric unit, friends, we people compulsory require strong acids only. So friends, example I am giving you. You take a grape. Outer coat of the grape, that is also tannin only, and whatever the fleshy part we eat, that is also grape only. Answer, that is also tannin only. There is a seed, we will break the seed, we will get a powder, that is also tannin only. What I would like to say here is tannins are basically exhibited more than 20 different forms. Now among the different forms, all the forms are broadly categorized into condensed and hydrolyzable. Condensed are made up of the same monomeric unit. That's why if we want to break, we should always use the strong acids. Hydrolyzable tannins are made up of two different monomers. Hence, by using dilute acids also, we can easily break them. Kindly do not forget the statement at all, friends. Okay, that is about the second class of the secondary metabolite, that is the phenols. Okay, friends, everyone say yes or no. Is that clear, everyone, till phenols? Any kind of a doubt in the phenols, I will re-explain. Please let me know. Okay, next and last and final one, the nitrogen containing compounds. You know, friends, what they are doing is from 2019, 2020, 2022, if you look into the question from secondary metabolite concept, how they are asking is, they are asking the question by including all. I mean to say, they are not including only terpene there. I mean to say, they are not specifically giving the question from the only terpene, only phenol or only nitrogen containing compound. Instead that, what they are doing is, they are giving the question from all the compounds at a time. Okay, very good. Continue, good. So they are giving all the components together and they are raising this particular part C question. Okay, so you have to read every single information, friends. Every single information. What is the greatness of these topics? The topics like secondary metabolites, the topics like nitrogen metabolism. The greatness of these topics are, the topics are very small. What, my friends? The topics are very small, but the information is too much. So, and moreover, this concept these small concepts are not greatly concept oriented or subject oriented. They are generalized topics. So that's why you have to remember most amount of the information. Okay, the last and final class of the secondary metabolite is the nitrogen containing compounds. Everyone listen to me. They are the group which include all the secondary metabolites, friends, including containing the nitrogen. Friends, listen to me. From here, everyone listen to me for 20 minutes continuously. Nitrogen containing compounds are broadly categorized into alkaloids, cyanogenic glycosides, glucosinolate, and the fourth one we can call non-protein amino acids. This is the fourth one. Friends, so far from the cyanogenic glycoside also we got a direct part C question, and friends, from the glucosinolate also we got a direct part C question. Please kindly don't forget that. Now friends, let's talk about the first one, that is the alkaloids. I can say to all of you, alkaloid is the largest class of nitrogen containing compound. Right now, whatever the table that I am going to explain you, friends, it is utmost important table, utmost. Okay, so we can expect a direct questions. I'm requesting all of you to remember that slide. Yeah, as I said it to you, friends, alkaloids are the largest family of the nitrogen containing compound. You know, if you look into the structure of an alkaloid, everyone listen to me, friends. If you look into the structure of an alkaloid, nitrogen will be present in the heterocyclic ring, some or other position compulsory. Nitrogen present in the heterocyclic ring. See here, some or other position, friends, there is a presence of nitrogen compulsory. Never ever forget that. That is the reason only we can call it nitrogen containing compound. And friends, basically all these alkaloids, I want you to remember, friends, all these alkaloids are generally derived from what, friends? They are generally derived from the common amino acids. Please kindly do not forget this point. Okay, so friends, this is the most important slide. I want every one of you to listen. You people no need to remember the structure. Apart from the structure, I want you to remember everything. Whichever I am going to mark, whichever I am going to explain you elaboratively, pay attention towards that. Alkaloid class, biosynthetic precursor, example, human uses. This is really interesting. Pay utmost attention, you will get a very nice idea. Pyrrolidine is the name of the alkaloid. It is derived from where? It is derived from the biosynthetic precursor. What do you call it as, friends? Ornithine. All the students listen to me. Pyrrolidine, it is going to give rise to what, my friends? The pyrrolidine is going to give rise to the nicotine. Never ever forget this point. Friends, what is the speciality of this particular nicotine? Listen, every one of you, listen to me carefully. Nicotine can act as a stimulant, depressant, tranquilizer. You know, friends, what do you mean by the word tranquilizer? We can call it mood changer. What do we call it? Mood changer, which can change our mood actually. Let me tell you. Suppose example, I am giving an example, friends. When a person smokes one cigarette, what the cigarette contain? Tobacco. What the tobacco contain? Nicotine. Once the nicotine release in the blood, nicotine will go and associated with the nicotinoid receptor. Once again listen, nicotine will go and associated with the nicotinoid receptors present in the hypothalamus. What they will do? It stimulate the nervous system. That's why it is called as a stimulant. But listen to me, whenever a person smokes one cigarette in 2 or 3 hours, once in 2 or 3 hours, he will feel greatly stimulated. But the same person smoking continuously 4-5 cigarettes in the span of 1 to 1.5 hour, in just 1 or 1.5 hour only he started smoking 4 to 5 cigarettes. Now it never act like a stimulant, friends. Instead of acting as a stimulant, it is acting as the depressant. Right. So what is the statement I would like to conclude? Every student listen the statement I am giving right now. Listen the statement. Any drug that act on the central nervous system at a small doses or at lower doses act as the stimulant. Any drug which is acting on the central nervous system at a higher doses, it can act like a depressant. You take coffee also. When we drink one coffee in the span of 1 or 2 hours, we will feel very stimulated. But if we continuously drinking 2 to 3 coffees in the span of 1 or 1.5 hour, we never get stimulated. It will create the drowsiness. So that's what you have to remember. Any drug which are acting on the central nervous system at low doses act as a stimulant, high doses depressant. What is the tranquilizer I told you? Mood changer. Correct na? When a person smokes the cigarette, he or she feels that they got relaxed. So they will come out of situation. So that's what we generally use a word mood changer or the tranquilizer. Do not forget that, friends. Whichever I highlight, never ever forget. Next one, the alkaloid tropane. Tropane is the alkaloid derived from the biosynthetic precursor ornithine. What are the examples, friends? Examples are atropine and cocaine. Friends, please listen to me. All the information is important to remember. Atropine prevent intestinal spasm. Friends, what is spasm? Conduction, muscle conduction. See, every student listen what I am showing to you. See, our muscles are like this. Our muscles are like this, keep on moving, keep on moving, keep on moving. You know, sometimes what happen, muscle will become like this. It's keep on moving like this, but muscle, sometimes it will become like this. We can call what spasm or conduction. So by that it create unbearable pain. So the atropine can used as a powerful drug in the prevention of intestinal spasm. We can call it muscle spasm or muscle conduction. Sometimes due to the gastric condition, we will get stomach muscle spasms also. That's why we'll get unbearable pain in the stomach, right? That is also what we can call stomach muscle spasms. Basically, we will use atropine during that particular time. And friends, another very interesting point, atropine can act as an antidote. What, friends? Atropine can act as an antidote for other poisons. You should not think that atropine act as a powerful antidote. That is wrong statement. Atropine, please listen to me. Atropine is one of the composition in the universal antidote. I hope you are getting me correctly. Atropine is one of the composition in the powerful antidote. Actually, antidote is made up of 3 substances. In the 3 substances, atropine is one of the substance. Simple point. That's why we can say antidote to the other poisons like snake venom, bee venom, for those. And atropine, friends, it's very rigid. I want you to remember. Atropine can also used for dilation of pupils for examination. Dilation of pupils for examination. I don't know whether you performed this kind of experiments or not. If anyone study the pharmacology, you might perform this. You know, we will take the atropine eye drops. We will take the atropine eye drops. We will put 1 or 2 drops in the eye of the rabbit. We will take the rabbit and we will put 1 or 2 eye drops of the atropine in the rabbit eye. Then we can easily understand the pupil movement inside the eye. So that is what we can call dilation of the pupil for the examination, where atropine can be used as the eye drops. Very simple, friends. Then the next and most important one, cocaine. So where we use this particular cocaine, all of you understand, cocaine can act as a stimulant of the central nervous system. I told you, right? and local anesthetic. Friends, every one of you listen my words carefully. What are anesthetics? I hope every one of you remember. Anesthetics are the drugs which make the body to lose its sensation. Anesthetics are the drugs which makes the body to lose its consciousness. Just listen. There are two type of anesthetics are there, friends. One we called general anesthetics. General anesthetics. Second one we called local anesthetic. What are general anesthetics? Just imagine. Suppose example, I am undergoing a heart surgery. So during the heart surgery, it is necessary that my entire body has to lose the sensation. Entire body has to lose the sensation. So that time what happened? Doctors will give general anesthetic. During the labor time, during the baby delivery also, doctors always will give the general anesthetics to the woman. Suppose example, I am doing a small surgery for my little finger. I am doing a small surgery for my little finger. Is there any necessity to take the general anesthetic and to make the entire body to lose the sensation? Not at all. It's not required. So during that time what we will do? We will take a local anesthetic. Local anesthetic means the function is common. They will make that particular part only to lose the sensation. So if I give the cocaine here, cocaine will act only on this finger. That's all. So cocaine can act like a local anesthetic which can helpful in losing the sensation of a particular body, particular part. So I want you to remember pyrrolidine, tropane, nicotine, atropine, cocaine. Next, the two very important ones are very important ones. Isoquinoline, indole. Okay, if you can't remember this, that's okay. Leave it. But if you remember, I really appreciate. Okay. If you remember this, remember. Otherwise, no problem. But pyrrolidine, tropane, isoquinoline, indole. These are all very important. Indole alkaloid class indole alkaloid class by using tryptophan amino acid as a precursor. You know, right? Tryptophan amino acid having what kind of ring? Aromatic ring. So, sorry, not aromatic ring, I'm sorry. It is having a indole ring. Now, this indole alkaloid, by using tryptophan, it is giving rise to the psilocybin, reserpine, stretchinin. I want you to remember. Psilocybin, it can act like a powerful hallucinogen. Friends, you know, this particular psilocybin, even it is present in the mushrooms also. Even it is present in the mushrooms also. I think my picture is covering all these three lines, but I am explaining you, just listen. Psilocybin is the one which can act like a hallucinogen. You know, when people consume mushrooms which contain this chemical psilocybin, listen, when people consume the mushrooms which contain this chemical psilocybin, they will undergo the hallucinations, means supremacy happiness. They are not under the control of their willpower. They are thinking beyond their limits. And please kindly don't think that this particular psilocybin containing mushrooms are not found here. They are most commonly found in the African forest. Then reserpine as a powerful drug which is involved in the treatment of the hypertension. It's one of the major drug of the choice. Whereas friends, stretchinin, very important one friends, stretchinin, which is used in the treatment of rat poison, in the treatment of eye disorders. I want you people to remember this without fail. Remaining, the only class is left, isoquinoline. I will cover that in the next class. Okay. Friends, this table seems to be very small, but it's very important for the part C questions. I'm requesting all of you, whichever I have explained, please kindly remember that without fail. Okay friends, that's all for now. I will meet you in the tomorrow class. And I'm informing you right now, from the tomorrow onwards, you will be having two classes. As I told you now, I will be covering the unit 7 also, unit 4 also. So I will be sharing you the timetable in the Telegram group. Don't worry at all. Okay. But we have two classes from tomorrow. Okay. Okay friends, that's all for now. Okay. I'll see you in the tomorrow morning class. Okay, bye all of you, thank you so much. Okay, bye, have a good day to all of you.